All right, well, welcome everyone who has joined us here. You've been hearing the voices of Mark, and do you go by Mike or Michael?
Mike is fine, yes.
And Mike, they are in the same room physically, but they are coming to us virtually from two separate Zoom interfaces, and we are very excited to hear what they have to share with us. So without further ado, gentlemen, we'll hand it over to you.
Thank you very much for this opportunity. We really appreciate this possibility to speak with you today. A little bit about product development cycles for medical devices and engineering. My name is Mike Kopcak. I manage the medical education team here at PRL at Medtronic, and I'm also a technical fellow. And I'll hand it over to my co-presenter, Mark, for his introduction.
Sure. So my name is Mark Palmer. I'm in our corporate core technologies group, and both Mike and I are part of an organization that's called Strategic Scientific Operations. So we have the opportunity to collaborate across all of the Medtronic enterprise on a number of projects and a number of different devices.
Yes. So hopefully you'll find our presentation insightful and helpful to inspire you in your own careers. Oh, we have a little glitch. Oh, here we go. So Medtronic is a medical device company, and it's developing therapies and treatments to address over 70 different ailments in a variety of areas such as heart disease, spinal disease, lung disease, and such. And so we'll talk about some of these products as we go through the presentation. So Medtronic is organized into 4 portfolios or groups, we call them: the cardiovascular, the medical surgical, the neuroscience, and diabetes portfolios. But what's nice, as Mark alluded to, is that we're in a unique opportunity to collaborate across all of these units, share wisdom and insight with each of these.
So just to give you a taste for some of the things that Medtronic makes, some of the devices that we produce, they're shown on this slide. In the upper left-hand corner, we have things like artificial heart valves. We have pacemakers moving across the top row. In the next row, we make devices like pumps and sensors related to diabetes. And then in the third row, there are devices related to surgical procedures. So things like staplers, endoscopy pills that a patient can swallow and it will image their digestive system. We do have robotic systems, which may be of interest to some of you that are participating in FIRST Robotics. We have imaging systems. And then in the bottom row, we also have devices for treating things like stroke, for treating orthopedic injuries, especially of the spine. And then we also have a robot that helps surgeons do implantation procedures along the spine. So I wanted to, as part of just kind of warming up to the idea of medical devices, I wanted to share with you or discuss with you some of the things that we think about as we talk about engineering devices for healthcare. And this relates a lot to robotics and autonomous systems. So robotics are machines that are capable of carrying out complex tasks by themselves. And autonomous systems are able to do that sensing and the processing and then automatically responding without external control. And as a medical device company, we try to get our devices to be able to do those things as well. So if a person is having a problem with their heart, we want to give them a device that they don't have to think about clicking every second to make sure their heart is beating. We want that device to be able to sense when their heart needs to beat and to be able to respond automatically so they don't have to think about the problem that they're having with their heart. So this relates closely to the biology because organisms are also these independent systems, and they're capable of responding automatically to their environment. And as we create these devices, it's this interplay between the mechanical device and repairing the behavior or the injury to the body that we want to become completely autonomous. The patient can manage, not have to think about managing their disease. Sorry, a little more technical difficulty on our side. Okay, there we go. So when we think about healthcare, what we're trying to do is optimize a person's health. And we want to be able to predict bad things before they happen so that that person can see a clinician or see a doctor. And by doing it this way, it will make the things that the doctor needs to do for them less intense or less severe, perhaps in avoiding surgery. And in order to do that, we have to be able to understand the inputs to the person's body and then predict what the outcome is going to be. So in the bottom left-hand corner of the slide, some of these inputs are like, what's their diet, what's their exercise, how are they sleeping, where do they live? And if we are looking at what happens in the hospital, then the things in the blue box come into play, like what medications do they take, what surgeries have they had, or what devices have we given them? But all of this is the goal is to be able to predict the impact on the person's body, whether it's their brain and nervous system, which is the sensors, their cardiac system, their digestive system, their musculoskeletal system. Those are like actuators, or their vital signs, or things that are happening in their blood chemistry. So can you advance again? So in order to get these products to the market, we provide 4 types of evidence. One is the experiments that we do on a bench. These are called benchtop tests. We can also do experiments in animals. We can also do what are called human clinical trials. So once we know that the device is safe, we want to see whether the device also provides benefit to the patient. And then finally, there's modeling and simulation. So moving to the next slide, we can see that with these 4 different ways of doing experiments or understanding the behavior or the relationship between the device and the body, not all of them are created equal. So animal tests have certain advantages, certain strengths and weaknesses, as do these benchtop experiments, the clinical trials that we do in human patients, as well as human simulation. And part of the goal is to put together these different types of experiments and tests in order to answer all the questions that are critical for us understanding how that device will interact with the patient long-term. So we're going to turn things back over to Mike, and he's going to focus on the animal studies and the work that gets done at the Physiological Research Lab.
Thank you, Mark. So Medtronic was founded by 2 gentlemen, one of which is Earl Bakken. And in the early 70s, Earl Bakken wanted to create a research center that would allow the engineers and scientists to go from the benchtop to the bedside. And so he had this idea to develop a research center that would support all of the preclinical or testing before the device goes into a human on site. And so as a result, PRL, or the Physiological Research Laboratory, was established. And our goal today is to support Medtronic's preclinical research and education needs through a variety of different mechanisms, all focused on quality, ethics, and proactive partnerships. So it really reinforces that collaborative innovation that we talked about a little bit earlier. The ethics piece is very important because, as we'll talk about in a little bit, we're very honored and privileged to be able to use animals and human cadavers to support our research efforts. So we'll talk a little bit about that in the future too. So the PRL, or the Physiological Research Laboratories, has 3 primary services that we can offer Medtronic. The largest is preclinical research. This is the development of animal and cadaver models that are able to test and quantify the safety for regulatory submissions for all of our new products and therapies that are being considered for human use. The biomaterials evaluation qualifies the materials and biocompatibility testing for the products or the components of our products. We want to make sure that those components are safe and they're not going to leach something that might be carcinogenic or toxic to the patient. And then finally, we want to make sure that clinicians understand how to safely and effectively use our therapies to treat patients. So we spend a lot of time making sure that they have the proper education and training to implant our devices and therapies. We have on our program an Institutional Animal Care and Use Committee, or an IACUC. This is mandated by federal law, but essentially you can think about this as an ethics committee. So anytime any of our scientists and engineers want to conduct a study involving the use of animals, this committee is reviewing all of those protocols to ensure that the animal use is appropriate and that we're actually going to benefit mankind by these animal sacrifice. So just to put this into context, some animals can save up to 600,000 lives. Animal research in general adds about 17 years to a person's life. So it is a very important part of this whole process. But that being said, we don't take the responsibility of using animals very lightly. This is very, very challenging to get this type of approval to do this type of research. We also have members of the community on our review board. And the purpose is to make sure that Medtronic isn't rubber stamping all of these protocols because it would be financially beneficial for us to bring this new product to market. We want to make sure that we're being check and balanced by these community members. At PRL, you can think about it as a small hospital. We have 7 surgical operating rooms, a human tissue lab where we can do various procedures. Much like a hospital, we have a variety of imaging capabilities, such as CT scanners, MRI, ultrasound, X-ray, anything that you would find in a hospital, we would be able to execute here at PRL. It's very important that as we design these products, that we're testing them in the environment that they'll be used. As I mentioned, we also do physician training. So we have training facilities that allow us to train our physicians on our latest therapies. We also have anatomage virtual dissection tables. These are a nice substitute for using human cadavers for studying anatomy or even doing virtual fitting that we'll show you in a little bit. We do have a lot of animals on site. We have primarily large species animals like sheep, pigs, dogs, and rabbits. And depending on what type of study we're going to do, we choose the appropriate model based on regulatory requirements and literature precedents. We also have the advantage of having anatomical and clinical pathology on site. So we're able to not only use our veterinary surgeons to implant the devices, but also use our pathologists' expertise to evaluate what changes are occurring in the body as a result of those therapies. As you can see, we also have biomaterials evaluation. So we have a lot of chemistry labs that support toxicological and chemistry assessments of those components. And then because we will bring in large groups, either for training or for research purposes, we have a variety of conference room space. This slide just shows you some of the high-level steps that are necessary during the product development process. So if you look at the left upper right-hand corner, you can see this is where ideas start. New therapies will start as early concepts. We'll start to think about what materials we might like to include based on needs requirements. We'll start thinking about what animal models or cadaver models we might need to test that new technique or therapy in. And then we'll start to do early feasibility testing. These are generally very crude devices that are built on the bench. They're not to the standard that you would implant in a human, but they give us an idea directionally of how that therapy is going to play out. And then we start to select, based on outcomes of preclinical studies, which of those concepts are most likely to be viable. And so then we move down the line into things like procedural development, like how is this therapy going to be delivered? Is it going to be on a catheter? Is it going to be open surgery? And that also then leads into some of the design features that are incorporated. Then we really start working on the preclinical studies. The definition of preclinical can vary, but at least in this context, we're going to include anything prior to human use. So that can include things that Mark's going to talk about later, like computer simulation, can include animal studies or cadaver studies. But during this whole process of preclinical studies, we're going from early feasibility into assessments of efficacy. We want to make sure that what we're developing is actually going to affect some sort of positive change. And then finally, we get into device safety, because we want to make sure that not only is the device going to be efficient and effective at meeting its end goal, but we also want to make sure that it's not going to harm the patient. Then we generally get into procedural optimization. So we'll bring in clinicians or other experts to help us understand how we can best deliver this therapy. And then we'll start to do field training for our field staff that support clinical cases, as well as our physicians. And usually around this procedure optimization time, we start going back up to the top and looking at the second generation of devices. So Mark and I thought it would be a great idea to show you a real life example. So we're going to talk a little bit about the micro transcatheter pacing system. And so this is a revolutionary new cardiac therapy designed to treat bradycardia, or a slow heart rate. So if you look here, this is the typical implantable pulse generator that was used in most cases. It has a lead here that's attached to the heart, and that lead is tunneled under the skin, usually up into the upper chest, and then an incision is made and this large device is placed under the skin. This device has all of the electronics, so it'll measure heart rate, it has the battery, it will deliver the therapy as the heart needs it. But this new micro, by comparison, is quite small. It doesn't have a lead. It's implanted directly into the heart. And, you know, it's attached by these tines here. So it's definitely a revolutionary thing. It was 93% smaller than this traditional pacemaker. So it eliminates a lot of complications like pocket infection, or even something known as Twiddler syndrome, where people will play with the device in their pocket and flip it over, which leads to fractures in this lead here. So it is really an amazing device. And what's also amazing is that it has a battery lifespan of about 8 to 13 years. So you'll see how small this device is later in the presentation, but just keep in mind how much engineering marvel goes into developing such a powerful battery. So here we're going to see a video. This is the delivery system. The micro is placed in a little capsule here in the delivery system. This delivery system, you'll see. And yeah, so here's the delivery system coming out of the introducer. And then the clinician is going to use the handle and make some twists and turns and put the micro into the intended target location. So that image that you just saw was basically a moving X-ray. So it's kind of cloudy, kind of hard to see. And but that's the state of the art right now in imaging. Here you can see the microtines have engaged in the myocardium, or the heart muscle, and they're kind of like fish hooks. So it's a material called nitinol, which is very flexible, and it takes the shape of that capsule here. And here you can see it engages with the heart tissue. It doesn't pull out. This is a perfused cadaver. So we're able to see the engagement of the device in the heart. And then, you know, the next step is just to test the electrical effectiveness of the implant. We want to make sure that we have a good capture of the heart and that we're delivering therapy effectively without over-consuming the battery. And then the final step is to cut the cord and remove this tether that holds the micro device to the delivery system. And then some pressure is held on this venipuncture site, and that's it. It's a very straightforward procedure that is quite revolutionary. These are some examples of some of the research work that we've done here at PRL. So here you can see some human cadaver preparations. So we've taken the fresh human cadaver that's not been embalmed or fixed, and we've flush the vascular system of any blood or clots, and then fill it up with water, and then we put videoscopes inside the body. And so we're able to see the delivery of the device. We can see the tines engaging here. You can see these tents. That's where those tines are engaging with the heart. But this is a much better view than those fluoro images. So the researchers and engineers are able to get a lot of good data The clinicians are able to have confidence in the transit of this device through the heart and improve their implantability. This is also just another example of some of the use of newer technology to help support development and education here at PRL. So this is an example of mixed reality using the HoloLens from Microsoft. And here, some of the designers are looking at the implantation of the Micra in a virtual heart. They're walking around the heart to see how it interacts with the tissue. And then on the right-hand side, you can see an exploded view or expanded view of the Micra device itself. So we can see how all of the various components are put together to create this new Micra device. These are some images that we've taken from our anatomage table. So we've imaged a cadaver on the right and then a human patient on the left and reconstructed these images using this technology so that we can appreciate how those different devices interact with the body. So this is a new device that Medtronic's made. It's called a extravascular implantable cardio defibrillator. So we're looking at the position of that lead here. Here we can see a neuro device implanted near the spinal cord. And then this patient also had previous surgery. You can see chest wires closing the sternum and a heart valve in place. So this allows researchers and scientists to see the technology implanted or the device implanted and what type of relationship it has to the surrounding anatomy. This is another example using our anatomage table, which is that virtual dissecting tool, where we've taken an image and rendered it in 3D, but then we've also implanted a virtual Micra using an STL file. And so this allows us to look at how the engineering marries with the biology. We can look at things such as how much anatomy is interfering with that device once it's in the target location, or even how many of these devices can we safely implant in the ventricle before we run into some challenges with obstruction of things like the heart valves. So it's a very, very powerful tool to help us understand during this product development stage what's going on inside the body. This is just another example of physicians being trained. So on the left-hand side, you can see the fluoroscopic image. This is what the clinician's going to see in their hospital when they're implanting in a patient. So it's a very grainy image. If the tissue doesn't have the correct density, the clinician's not going to see it because it's not going to pick up under X-ray. But with the videoscope, they're able to see those structures that are invisible under fluoro and really help improve their implant technique. So if you think about it, that translates directly to improved patient outcomes and reduced complications during the procedure. And just for some levity, I thought I'd introduce just some fun facts about our animals. We use a lot of positive reinforcement to encourage our animals to do certain tests or tasks. And so we'll also use them to deliver medications. We go through a lot of marshmallows, about 1.5 million calories worth of marshmallows, or about 68,000 marshmallows in a typical year. We go through about 50,000 fig Newtons, and believe it or not, the animals actually have a flavor preference. They like the reds, such as raspberry and strawberry, over blueberry or fig or apple Newtons, just in case you needed to know. And we also go through a lot of hay. Now, I'm a city boy, so this numbers boggle my mind, how much hay we go through, but even some people with a farming background say this is still a lot of hay. So about 71,000 pounds of hay in a typical year. And with that, I'm going to transfer it back over to Mark, and he's going to talk about some of the in silico testing that we do with engineering and biology.
Thanks, Mike. So you saw some good examples of just how important collecting different types of data are to the entire engineering design process. And I think at the end there, you also saw how important things like video, rendering, all those technologies that go into video games, they're becoming more and more important to being able to see and visualize and understand what's happening in the clinic or in the surgery. So behind some of those technologies is modeling and simulation. So we're going to talk about that now. And this slide is a slide from the Food and Drug Administration that does all the approvals for medical devices. And it identifies all the different ways that modeling and simulation, those physics engines that underlie video games, play an important role in the medical device industry. So in the top row, we can see the more of the design and development process. So simulating the device, simulating the anatomy, understanding the physiology, and then some of our products actually deliver pharmaceutical agents, so understanding drugs and how those interact with the body, and then also being able to simulate how our devices are manufactured. And then once the device is kind of in the hands of the clinician or in the hands of the patient, there's modeling and simulation that's running inside the device as part of the control systems. We can also help the clinician by simulating some of the measurements that are taken in the clinic. In this case, that's called simulation as a device. And then we can also simulate treatment outcomes. We can use simulation to screen different types of therapies or different ways of managing the patient. And then with AI and machine learning, we can combine many different data sources to simulate how the patient will do over time. So on the next slide, you can, I'll just talk through where there's opportunities for growth in modeling and simulation. So this modeling and simulation technology comes from aerospace and automotive and energy industries for things like crash testing or aerodynamics of race cars and things. And those technologies have helped us be really good at simulating the physics of the device and how it's going to behave under different loads. Where we are growing as an industry is understanding what happens when the clinician is trying to put that device in the body, as well as how that device gets loaded when the patient has that pacemaker and then they go out and they do weightlifting or they go cycling or they go jogging. What does that do to the device? And then finally at the top there is anatomy and physiology and the simulations for understanding how the body is going to interact with that device over time. So if you've ever had something stick in your finger or anything like that happen to you, you know that your body doesn't like having things that aren't part of your body stuck inside it. And from the moment that happens, it starts to wage a war against it. We need to understand that process because medical devices are not natural biological things, and the body is going to be in this battle against them for as long as they're in the body. So these are areas where we're trying to combine different modeling and simulation technologies with the idea that as we get better at modeling, then we can rely less on testing these different approaches, these different technologies in animals or in humans, and be able to really do a lot more exploration in the computer. So transitioning to the next slide, this is kind of the modeling and simulation landscape for that micro device. There is a lot of different styles of models that are taking place. We need to understand energy and how it's being used by that battery so we can make it last for 8 to 13 years. There's a lot of circuit design in there to optimize the use of energy and the communication between the device and the outside world. There's a lot of physiology that is modeled in the system as well to understand how to interpret the electrical signals that are coming from the heart and for the device to know when it needs to intervene. And one of the things that people don't think about is that when a person gets injured for another reason and they need to get an X-ray or a CT scan or an MRI, that can interfere with the device, and we need to understand that as well. So I'm going to skip down to, can you jump to slide 40? Mm-hmm. Or no, slide 39 is good. So got one more.
No problem.
Yeah. So in the upper right-hand corner is a movie that is collected from one of our animal studies that was performed at PRL. And Mike mentioned the little hooks that anchor that device in the heart. And this is actually a CT scan of the micro device implanted in an animal heart. And we need to understand things like what's going to happen to the device when the heart is beating. And your heart beats around 100,000 times a day, which means that these little hooks are going to get flexed that many times. So we need to be able to take findings like this and convert them into experiments and not wait 8 years or 13 years to collect that data. Can we accelerate that test so that we can answer that question in a week or a month about whether the device is designed sufficiently to handle that many heartbeats? If you move to the next slide, then we can also look at how that electrical signal from the device activates the muscle in the heart. And then when that muscle contracts, how it squeezes the blood out of the heart, and is it creating the efficient contraction that mirrors what a normal heartbeat would look like? Another design question on the next slide is the interaction between the device and the anatomy. And the heart is full of blood vessels that supply nutrients to that cardiac muscle, and understanding what the chances are of one of those little hooks that anchor the device to the heart muscle. Will it interact with one of these vessels and injure the heart? And the answer to that question is no. But we had to answer that design question before putting that device in a patient. So moving to the MRI problem, when you put metal in an alternating magnetic field, which you may have been exposed to in your physics classes already, it will cause a current to flow. And that can interfere with the beating of the heart. And we needed to be able to answer that question of whether an MRI machine will cause the micro device to pace the heart in a bad way. So the way we answer that question is what's called hierarchical validation, or doing validation in stages. So we start with the benchtop test and then progress to an animal study until we do a simulation of the device in a human. So this progression is shown in the simulations that are shown on the left. And on the next slide, this is all the variability that goes into a model like that. So starting clockwise at around the 4 or 5 o'clock position is a simulation of the scanner. And then we have a simulation of the different ways the patient can be put inside that scanner. And then up at the 2 o'clock position is different sizes and types of patients in the scanner, different design leads, different lengths of leads, and then finally differences in the path that the lead took when it got into the patient. So when we put all of that together on the next slide, we can answer this question of what are the odds of something bad happening? And as you can see, those odds are really small. They're 1 in 75,000 to 1 in a million patients may experience an accidental heartbeat coming from the device due to the interaction with the MRI scanner. So the last slide that I wanted to share kind of on this progression of modeling and simulation in medical devices is looking at modeling and simulation across the clinical workflow. So we need to identify which patients have a disease. We need to understand their diagnosis. Then we need to do the planning for how we're going to treat them. We need to carry out the procedure, and then we need to know whether the procedure was effective. So underneath are these different applications of modeling and simulation. So there's modeling and simulation of screening technologies for things like breast cancer. There's modeling and simulation to simulate the placement of a stent in the cardiac vasculature and understanding how that stent will interact with a plaque in that patient's diagnosis. In the pre-op planning, we can do different sizes of devices using simulation to understand what is the best fit and to help with the planning of the procedure. Intraoperatively, for things like surgery and robotics, we can take the image of the patient, the device that we want to put in, the different screws that are used to anchor that device to the patient's bones, and plan all of that, program it into the robot, and the robot will guide the placement of the tools and the screws that are used in that procedure. And then all of that data can be used to analyze the outcome of that patient following the procedure to help us understand how to improve it. So we're going to close with...
Other than knowing that I have the micro, I don't know that I have the micro. My name is Steven. I'm 39 years old. I'm a singer-songwriter. I like up close and personal back porch kind of stuff, but I still like to strap on my guitar and run up and down the stage and act a fool from time to time. In the summer of 2014, I was driving my truck down the interstate. I remember feeling dizzy. I had the motion sickness, and the next thing I know, I've blacked out and slammed my truck into the side of a bridge. I was diagnosed with having sick sinus syndrome, which is a condition where I have messed up electronics in my heart, and it allows my blood pressure to drop to a point that my heart stops. I had just gotten married and, had chosen the person that I wanted to sit in a rocking chair with. My kids are reaching an age where I start considering the possibility of grandkids, and now this is being jeopardized. When my doctor presented me with the Micra as an option, it was a real easy sell for me. He said, "You're not going to have anything visible. The recovery time is a fraction of the time that you would have with a traditional pacemaker." At the time that I had the surgery, I was actually in the middle of a workout regimen. I remember asking my doctor, I was like, "Well, look, when can I get back into the gym?" He said, "Look, as long as you're not doing any heavy lifting," he said, "as soon as you feel like it, get after it." I had the surgery on Thursday, and that next Tuesday, I was back in the gym. I'm absolutely amazed something so tiny can play such a huge part in maintaining quality of life, physical and psychological. That little tiny thing right there is going to allow me to live a long, full life right there.
All right, so hopefully with this presentation, we want to give you a taste for the things that you're doing right now in FIRST Robotics, things that you're learning in school, and how robotics and autonomous systems are useful in the things that you're going to be doing for your FIRST Robotics competition, but also how they map later on into industry and into things like healthcare. So with that, we're happy to answer any questions you have.
I'll go first. Oh, no,
Okay.
go ahead. Go for it.
I was just curious, out of all the things you've talked about that you developed and designed and everything out of those softwares, some of the things that didn't quite work out and didn't go the way you wanted them to, did you have a way to reuse those and they turned into something new that you hadn't expected before?
That's a great question.
Yeah, fantastic question.
Very, very good question. Mark, do you want to go first, or do you want me to go first?
I'll let you start.
Sounds good. Thomas Edison was once asked if he felt badly for all of his failures when he was trying to invent the light bulb. And he said, "No, not at all. I learned 10,000 ways that something doesn't work." And so, I think we take that same approach, especially with the work that I do. A lot of it is preclinical. It's using a biological system like an animal. And what I always tell people is, the animals and even patients, they don't always read the same books that we write. And so there's a lot of last minute changes, a lot of, "Oh, I wasn't expecting that," or really making you think about what is confounding your data. There are some things that we just didn't anticipate. But then there's also, like you said, probably some serendipitous benefit that arises. And, I can't think of anything off the top of my hand where, we've been able to recycle very quickly, but there have been some models where we wanted to create, for example, a calcified aortic leaflet, where it represents a leaflet that you would find in an elderly person. And so we did models where we did all of the things that we're told never to do when you're doing cardiac surgery, like touch the leaflet with your forceps or causing some sort of abrasion, to try to stimulate the body to have these types of responses. So it's kind of using some of those outcomes in a way to get what you want. But I think still anything that we learn due to our failures, we incorporate, and it goes into improving the reliability of the outcome.
Yeah, and just to build on that answer, I mean, we were talking about that micro device that was developed over decades. There were questions like, how are we going to power this device in the body? And they talked about nuclear energy. They talked about all kinds of things. And we failed until we were able to advance battery technology to the point where we could have these small batteries at the size that would fit into this device and still be able to meet the energy needs for the patient. Similarly, with the anchoring mechanism for the device, going through many design changes to get that device to be able to interact with the tissue well and to hold on to it without injuring it. There was a lot of failures there. But the cool part is that along the way, you develop these technologies, whether it's imaging or modeling or surgical techniques, that allow us to learn more rapidly and get more insights with each failure, so that we can design more rapidly and more safely.
Right. And I think a lot of people think that failure is a bad thing. It's not. As long as you're learning from your failure and incorporating it, it's actually quite helpful, because it's much better that it fails when it's in my hands or Mark's hands rather than the doctor's hand implanting it into your grandmother.
Wow, that was such a great question. Can I get your name, whoever asked that question? I can't see your name because you called in.
That was Natalie.
Natalie, thank you so much, Natalie.
All right, I've got a question.
Yes, please.
In FIRST, there's a principle of cooperation, which is, teams should help and cooperate with each other even when they compete. The PRL, the Physiological Research Lab, do you allow competitors to use the facilities, or do you work with institutions or other groups to cooperate?
Generally, it's a mixed answer. So when it comes to things like animal welfare, or sometimes even employee development, we do collaborate. So for example, if there's an outbreak in a particular animal disease, we do talk to the other veterinarians at the competitors. But we don't generally collaborate with the competitors on direct product unless there's some sort of strategic reason that's occurring in a different area, well above what Mark and I would approve. But it is a challenge, because a lot of the institutional knowledge stays within the institution compared to an academic setting where there's a greater emphasis on publishing results. So it's a little different, the way that we compete and share knowledge.
Yeah, and just to build on that answer, Medtronic and many of the medical device companies do a lot of public-facing research that fund like academic institutions, universities, and things. And it's to advance the state of the science in the area. Yes. When it tends to become more private is around our specific designs. So if we can share knowledge to make the entire field better, we do that. And we actually collaborate with each other on that. But when it comes to the specific things that we make based on that knowledge, then that's held a little more closely to the vest.
Wow, great, thanks. And where do you get your animals for your animal models?
That's a very good question. So all of our animals are coming from USDA inspected vendors. Some of the animals, such as our dogs, are purpose bred for research. So if you think about it, we actually improve the quality of the research by knowing the medical histories of all of these animals from birth, and we even know their lineage. So we're able to reduce some of the confounding variables if we weren't able to have a controlled source. Our more agricultural animals like pigs and sheep come from vendors that have a side production focused on research applications. But in all cases, those animals are, those animal facilities are inspected by the USDA to ensure that they meet all of the regulatory requirements for things like space, housing, feed, adequacy of veterinary care.
Hi, I had two questions, actually. The first being that when I first learned of the modern pacemaker, I also learned that it was like revolutionarily small.
Yes.
For the time. And obviously now this device is a lot smaller, and I was just curious, like how much smaller do you think pacemaking technology could get? And my second question was about how do you guys intend to remove and or replace the micro after it, after the 8 to 13 year period?
Sure. Do you want me to take the second one and you do the first one?
Sure.
Sounds good. So actually, there's a lot of different technologies that we can look at to remove some of the micros. So some of the research that we did initially, if you remember that one slide we showed the STL file inside the heart, we were looking at how many micros we can implant safely. And some people feel it's 2 to 3. So if you think about it, you could get potentially 16 to 23 years before you actually have to remove them. But if you're actually going to remove them, we're looking at some technologies, maybe even involving a laser that can remove the tissue that's holding that device in place that forms naturally as the body tries to encapsulate that foreign body. And then there are things called snares that we can grab onto the back of the device and capture it again and then bring it back into a tool to remove it safely from the body. So there's always those types of engineering feats that are being considered on how to get it out when it's necessary.
Yeah, as far as the size, there's actually two competing things at play there. So one of them is continuing the reduction in the size of the electronics. And if you are able to reduce the size of the electronics, you can reduce the size of the battery as well, and still have the same the same amount of functional life. So those are key technology areas, but also emerging is what if you could actually do something to help the body repair itself and you won't need the pacemaker. So medical device technology is also working on that front, on technologies to help engineer or help the body repair itself, to repair the conduction system, for example. And it'll be interesting to see how those two competing themes play out over the next decade.